Hello, my fellow cardiology enthusiasts. My name is Hirsch Doshi, and I'm currently a second-year cardiology fellow at Thomas Jefferson University Hospitals. Today, I'm fortunate enough to be sitting with Dr. Jonathan Piccini. He is the director of the Cardiac Electrophysiology Department at Duke University and published over 450 research articles throughout his career. Today, as part of the Fits on the Go initiative, we'll be talking about the PacificAF Trial, a trial which he's one of the lead authors in.
It's great to be here, having been a fellow, I think, not too long ago, and please call me John. I'm excited to be here and talk to you guys about PacificAF. Have you had a good meeting so far, Harsh?
Yeah, I think the meeting has been a tremendous opportunity to finally see people in person and just to interact and network and connect and learn about all the new innovation that's happening in cardiology. So I'm very excited for this entire weekend itself. While we're talking about innovation, the PacificAF trial, can you just give us a brief overview of the motivations behind the trial itself?
So it's no exaggeration to say, right, that atrial fibrillation is incredibly common, and certainly all cardiovascular health professionals, and I'd argue all internists, everyone, we struggle with atrial fibrillation. And one of the struggles we face is that we know that the vast majority of individuals with atrial fibrillation require some form of stroke prevention therapy. But we know that stroke prevention therapies have limitations, and one of the limitations they have is that they predispose to bleeding. And so very frequently, as many in older populations as 1 in 4 patients cannot tolerate long-term oral anticoagulation because of bleeding risks. And so sometimes people say, well, how could we possibly improve direct-acting oral anticoagulants like apixaban, rivaroxaban, edoxaban, dabigatran? And one potential way to improve upon them would be to try and identify pharmacologic agents that have lower risks of bleeding. And that's where the factor XI inhibitors come in. And so PacificAF is testing the hypothesis or unmet need that we could identify an anticoagulant that would lead to less bleeding risk than direct oral acting anticoagulants. And so we hypothesized that asundexian, which is a small molecule factor XI inhibitor, would lead to less bleeding than apixaban, which, as you know, is a very commonly used direct acting oral anticoagulant that is very effective and very safe. So comparing, best of class direct oral anticoagulation against these novel factor XI inhibitors.
Oh yeah, I think that these are very interesting points that you brought up, especially in patients who are more prone to bleeding than others. I think this is a good opportunity to test this not only in atrial fibrillation but possibly other thromboembolic diseases as well that patients need anticoagulation for. But speaking about the PacificAF trial, can you tell the audience a little bit about the methods behind the trial itself, and ultimately what everyone wants to know is there less bleeding?
So, just to back up a tiny bit, one question would be, well, why would you think that a factor XI inhibitor would lead to less bleeding? And the rationale for that is if you look at patients who have clinical factor XI deficiency, it's very interesting. They may have difficulties with bleeding after surgery, but they tend to not have a significant risk of spontaneous bleeds like intracranial hemorrhage or GI bleeding. What's also interesting is if you look at patients with factor XI deficiency as determined by genetic testing, what's interesting is that those patients appear to have some protection against vascular events. And so because of those observations, we thought factor XI inhibitors would be an interesting therapeutic agent. So in PacificAF, what we wanted to do is we wanted to test this hypothesis that factor XI inhibition could potentially reduce the risk of bleeding, and we want to identify a dose of asundexian that would lead to effective suppression of factor XI activity and reduce bleeding. And so we had 3 arms: apixaban dosed according to label, asundexian 20 milligrams once a day, and asundexian 50 milligrams once a day. We randomized 755 patients in a 1-to-1-to-1 strategy, and then followed them for 12 weeks for the primary endpoint of ISTH major bleeding and non-major clinically relevant bleeding.
And did you look at all patients, or were there any patients you excluded, like patients who are on end-stage renal dialysis, etc.?
Yeah, no, it's a good question. We had your standard exclusion criteria, and then we also tried to enrich. So we tried to identify patients who had increased risk for bleeding, like those with chronic kidney disease. Asundexian is eliminated 15% by renal elimination, and so it may have advantages in patients with chronic kidney disease as well.
Excellent. No, these are all great points, and I think now we're at the stage where what are the final results? We're waiting to know.
Yeah, I keep avoiding the answer. So what was really interesting was that Asundexian. Well, let me step back for a minute. I'll delay even further. So one interesting thing is in 12 weeks of follow-up in 755 randomized patients, we didn't have any episodes of major bleeding. But we did have episodes of clinically relevant non-major bleeding. And when we looked at that and all bleeding, what we saw is there was approximately a 50 to 60% reduction in all bleeding with Asundexian compared to apixaban. Now, you may say, well, what is the meaning of any bleeding event or clinically relevant bleeding? And I think for clinicians, we know that those are important bleeding events. They may not necessarily lead to death, but they certainly often lead to therapy discontinuation. And there's been some evidence in observational studies that those events correlate with major bleeds. So yes, there was a reduction in all bleeding. We also saw that Asundexian led to effective suppression of factor XI activity, such that patients had a reduction so that they only had, say, 5 to 10% of their baseline factor XI activity.
Okay. And do you know, in terms of the pharmacology of this factor XI A inhibitor, what is the half-life and how long does it last in your system?
Yeah, so it's always important to emphasize where we are in drug development, and so we're early. We believe the half-life in healthy volunteers is between 12 and 17 hours, and that's why it's dosed once a day. I should say, again, it's important to emphasize that PacificAF was a phase 2 study aimed at testing the hypothesis that Asundexian would lead to less bleeding and to identify a good dose to study in a phase 3 clinical trial. It wasn't powered to look at cardiovascular events like stroke or MI. That hopefully we will study Asundexian in a phase 3 study where we can figure out if it does reduce cardiovascular events.
You heard it here first. Asundexian. Keep an eye out on Asundexian. It reduces bleeding compared to apixaban, at least in the phase 2 trial. Looking forward to the next steps and the future studies coming out hopefully soon.
Yeah, and I'll say I think, just to expand a little bit further, this is a really exciting time to be learning more about atrial fibrillation and how we can treat patients. For the longest time, we've had a single class of oral anticoagulants, and then we had the DOACs. And I think in the future, we may have multiple pharmacologic options for stroke prevention, and we may have multiple device options for stroke prevention. And that's a great thing, not only for patients, but also for clinicians as well.
And sorry, I just have one more question to ask you, because we live in a world where we're trying to have more heterogeneity in our trials. In terms of the patient population, were they heterogeneous, or was it male predominance, female predominance, Caucasians?
Yeah, so it was a study predominantly conducted in North America and Europe, and so the population is commensurate with that. I think it is a really important point, and it's hard in a trial of 755 patients to have good evidence for heterogeneity effect across whatever factor we're looking at. But I think the advantage of these large atrial fibrillation trials in phase 3 is that we're talking about well over 10,000 patients. And so I think it's exciting to think about all the information we'll have as we continue to do large clinical trials in patients with atrial fibrillation.
Great. Thank you for taking the time and talking about PacificAF. We'll hope to see it live.